Hello everyone. Hello everyone. Hello everyone. In C3 plant, some oxygen does bind to rubisco and hence CO2 fixation is decreased. Here the RUBP instead of being converted to two molecule of PGA bind with oxygen to form one molecule of phosphoglycerate and phosphoglycolate. Two carbon in pathway called photorespiration. The photorespiratory pathway there is neither synthesis of sugar nor ATP. That's why it is known as a wasteful process. Rather, it results in the release of CO2 within the utilization of ATP. The photorespiratory pathway there is no synthesis of ATP or NADPH. The biological function of photorespiration is not known. In C4 plant, photorespiration does not occur. This is because have a mechanism that increase the concentration of CO2 at the enzyme site. This takes place when the C4 acid formed from the mesophyll is broken down into a bundle sheet to release CO2. This result is increasing the intracellular concentration of carbon dioxide. In turn, this ensures that the rubisco function as a carboxylase, minimizing the oxygenase activity. Now that you know that C4 plant lack photorespiration, you probably can understand why productivity and are better in these plants. In addition, these plants show tolerance to high temperature. Based on the above discussion, can you compare plants showing C3 and C4 pathway? Use a format table given and fill the information. I am reading the NCERT of class 11th. Reading the NCERT of class 11th. Now I am going to start the Calvin cycle. Calvin, hello. Calvin and his co-workers then work out the whole pathway and shown that pathway operated in a cyclic manner. The RUBP was regenerated. Let us now see how Calvin pathway operates and where the sugar is synthesized. Let us at the outset understand very clearly that the Calvin pathway occur in all photosynthetic plant. That this pathways occur in all photosynthetic plant. It does not matter whether they have C3, C4, any other pathway. For ease of understanding, the Calvin cycle can be described under three stages: carboxylation, reduction, and regeneration. My day was good. And the first process is carboxylation. Carboxylation is the fixation of CO2 into a stable organic intermediate. Carboxylation is the most crucial step of the Calvin cycle, where CO2 is utilized for the carboxylation of RuBP. This reaction is catalyzed by the enzyme RuBP carboxylase, which result in the formation of two molecules of 3-PGA. Since this enzyme also have an oxygenation activity, it would be more correct to recall it RuBP carboxylase, oxygenase, or Rubisco. It is the most abundant enzyme in the universe. The second process is the reduction. What is reduction? These are the series of reactions that lead to formation of glucose. The step involve utilization of two molecules of ATP for phosphorylation and two of NADP for reduction per CO2 molecule fix. The fixation of 6 molecules of CO2 and 6 turns of cycle are required for the formation of 1 molecule of glucose from the pathway. And the third is regeneration. Regeneration of the CO2 acceptor molecule RuBP is crucial if cycle is to continue interrupted. The regeneration step requires 1 ATP for photophosphorylation of the RuBP. Let us jump on the page number 218. Hence, for every CO2 molecule entering the Calvin cycle, for every CO2 molecule entering the Calvin cycle, 3 molecules of ATP and 2 molecules of NADPH are required. It is probably to meet this difference in number of ATP and NADPH used in dark reaction that the cycle photophosphorylation take place. To make 1 molecule of 6 over 6 of turn of cycle are required. Work out how many ATP and NADP will be formed. Okay, fine. So how many NADP and DP? How many ATP and NADPH molecule will be required to make 1 molecule of glucose through the Calvin cycle? It might you understand all of this if we look at what goes in and what comes out of the Calvin cycle. What goes and what come in? 6 CO2 and out 1 glucose, 18 ATP and 18 ADP, 12 NADPH and 12 NADP. Now let us jump on the C4 pathway. Plants that are adapted to dry tropical region have C4 pathway mentioned earlier. Though these plants have C4 oxaloacetic acid as the first CO2 fixation product, they use the C3 pathway or the Calvin cycle as their main biosynthetic pathway. Hence, then in what way are they different from C3 plant? This is a question that you may reasonably ask. C4 plant are special. They have a special type of leaf anatomy. They tolerate high temperature. They show a response to high light intensities. They lack a process called photorespiration and have greater productivity of biomass. Let us understand one by one. Study vertical section of leaf. one of the C3 plant and other of the C4 plant. Do you notice the difference? Do both have the same type of mesophyll cell? Do they have similar cell around the vascular bundle sheath? The particularly large cell around the vascular bundle of C4 plant are called the bundle sheath cell, and the leaves which they have a such anatomy are known as a crane's anatomy. Crane's means width and is a reflection of arrangement of cell. The bundle sheath cell may is a reflection of arrangement of cell. The bundle sheath cell form several layers around the vascular bundle. They are characterized by having a large number of chlorophyll, thick wall, impervious to gaseous exchange and no intercellular space. You may like to cut section of leaf of C4 plant, maize or sorghum to observe section of leaf of C4 plant. Crane's anatomy and the distribution of mesophyll cell. It would be interesting for you to collect leaf of diverse species of plant around you and cut vertical section of leaf. Observe under the microscope. Look for the bundle sheet around the vascular bundle. The presence of bundle sheet would help you to identify the C4 plant. If bundle sheet are present, that it will be a C4 plant. Now let us go towards there. Now study the pathway shown in figure number 13.9, the pathway that has been named Hatch and Slath pathway, because C4 cycle is given by a Hatch and Slath pathway. That's why it is known as the Hatch and Slath pathway. Hello SAZ Ziauddin, please like it. Is again a cyclic process. Let us study the pathway by listening the step. The primary CO2 carbon dioxide acceptor is 3 carbon molecule which is known as phosphoenol pyruvate and is present in the mesophyll cell. The enzyme responsible for the fixation is known as PEP carboxylase or pepcase. The important to register that mesophyll cell like Rubisco enzyme C4 cycle oxaloacetic acid form in the mesophyll cell. It then form other 4 carbon compound like malic acid, aspartic acid in the mesophyll cell itself, which are transported to the bundle sheath cell. In bundle sheath cell, these C4 acid are broken down to release carbon dioxide and 3 carbon molecule. The 3 carbon molecule is transported back to the mesophyll where it is get converted into the PEP again, thus completing the cycle. The CO2 released in the bundle sheath cell enter the C3 and the Calvin pathway. The pathway common to all plants, the bundle sheath are. The bundle sheath are rich in enzyme which is known as what? Known as ribosco, ribulose, bis, phosphate, carboxylase, oxygenase. And what is ribosco? Ribosco is the most abundant enzyme in the universe. And when we talk about the most abundant enzyme in the animal, so it is a collagen but lack peptidase. Thus, the basic pathway that result in the formation of sugar, the Calvin pathway, is common to C3 and C4 plant. Did you note that the Calvin pathway occur in all the mesophyll cell of C3 plant? In C4 plant, it does not take place in the mesophyll cell but does so only in the bundle sheath cell. Now let us talk about photorespiration. Hi, Aman Neat 01. What about your NEET preparation? Hello, Aman. NEET. So now talk about the photorespiration. Let us try and understand one more process that create an important difference between the C3 and C4 plant, which is known as a photorespiration. To understand the photorespiration, we have to know a little bit about the first step of the Calvin cycle. What is the first step of the Calvin cycle? It is known as the CO2 fixation step. This reaction where the RUBP combine with the CO2 and form the two molecule of 3-PGA, that is catalyzed by Rubisco. So now we read this topic. Yes, I am a dropper. All animal including human being depend on the plant for their food. Have you ever wondered from where plant get their food? So yes, if we thought about it that from where plant get their food, so the first question arises in our mind that the plant can eat food. No, plant does not eat food. Plant make the food by the process of a photosynthesis. Yes, Aman, I am a dropper. All animals, including human beings, depend on plant for their food, and we human beings are also depend on the plant for the food. Have you ever wondered from where plant get food? Have you ever wondered from where plant get food? Yes, the plant is an autotrophic organism and they make food. Good that you are in class 12th. Work hard. Yeah, it is good that you take a PCMB stream. I am a PCB. That's why I am facing a problem in solving physics and some physical chemistry questions. Aman, please subscribe to my channel and please hit the bell icon. Good. Please like my live session. Okay, fine. Okay, fine. Now we again is going to start. I'm from where? I'm from. Where are you from? Green plant, in fact, to have to make or rather synthesis the food they need, and all other organisms depend on them for their needs, such as human being and the animals. The green plant make or rather synthesis the food they need to through a photosynthesis, and therefore they are known as autotroph. You have already learned that the autotrophic organism is found only in plant, and all the other organisms that depend on green plant for food are heterotroph. Green plant carry out photosynthesis, a physico-chemical process. Yeah, it might be asked in the paper that which process does the photosynthesis belong. Have you ever ruined the situation? Yeah, I know about the re-neat and all. What was my favorite college? So, yeah, every neat aspirant dream for a aims. So, green plant carry out photosynthesis is a physico-chemical process, and it may be asked in the paper that which process belong it, the physico-chemical or physical process or a chemical process or psychological process. You have to put a physico-chemical process in which they use light energy to drive the synthesis of organic compound. Ultimately, all living form on earth depend on sunlight for energy. The use of energy from sunlight by plant doing photosynthesis is the basis of life on earth. Photosynthesis is important due to two reasons. There are the two reasons for the photosynthesis are important. So it will be asked in the question that there how many reasons photosynthesis are important. So the answer would be a 3, 4, 5 option, but you have to put on 2. Basis of life, photosynthesis is important two reasons. The first reason is the primary source of food for all on the earth. It is responsible for the release of oxygen into the atmosphere. And the second one is the release of oxygen under 4,000, 5,000 rank. Yeah, this belong from the general category, that's why. Oh bhai. So I am a dropper of it here. Yeah, have you ever wondered to what would happen if there was no oxygen to breathe? So if there were no oxygen to breathe, that only uh, this chapter focuses on the structure of photosynthetic machinery and the various reactions that transform light energy into the chemical energy. More than two drop. Let us try to find out what we already know about photosynthesis. Some simple experiment you may have done in the earlier classes have shown that chlorophyll is a green pigment of leaf. That's why I am saying to you that study Uh, a variegated leaf or leaf that was partially covered with black paper and exposed to light. Yes, Aman. You have to solve a lots of mock tests. That what I experience in my drop year. That which things are lack in my preparation. No, no, no. I didn't start from class 12th. After class 12th, I taken a drop and then yes, you get it if you work. But please don't lose your hope until you reach your goal. Aman, please like my video. You have to study hard. Then you have to solve all the PYQ and read each and every line of NCRT. Just like is it 2? So it will be a 2. And in option if given is a 3, 4, 5, that you remind this page, that in which page of the NCRT this line was written. This type of preparation is required for the aims. That what I experience. Yes, best of luck. And please don't waste time on these live session and all. Do study. Yeah, hello Shivam Kumar. On testing these leaves for the presence of starch, it was clear that photosynthesis occur only in the green plant of leaf in the presence of sunlight. Your second attempt, yeah, you also work hard and read each and every line of the NCRT for bio. And the for chemistry, you have to follow NCRT also. And the questions of the NCRT exemplar. According to my experience, that don't feel scared. NEET is not about a scare or anything else. If you want to be a doctor, just feel like a doctor. Sometimes I thought that I ruined my life myself. This live session is only just for me also. Just I want to revise the biology and also just to tell all the NEET aspirants that about that how they learn the biology, how they read the biology. We did not follow a basic routine. We have to work like a robot if we want to clear a NEET. NEET is only an exam, but okay, fine. Good. Yes. Oh, on NEET prep. Good. NEET prep is a very good platform for the NEET aspirant. The couples are there. For the question target batch. How many questions you solve from target batch? How many questions? Shivam, please like my video. I said how many questions you solve? The more than 10,000 or 4 5,000? 160 attempt now in 1 hour. No, not about the 160 1 hour. How many questions in NEET prep? That there was an option there that where you checked it how many questions you solve from NEET prep. from the target batch. Pencil. So we talk about the another experiment you may have carried out where a part of leaf is enclosed in a test tube containing a some KOH soaked cotton which absorbs CO2 while the other half is exposed to air. The setup is then placed in light for some time. On testing for the presence of a starch later in two parts of leaf, you must have found that the exposed part of leaf tested positive for starch, while the portion that was in tube tested negative. This shown that CO2 was required for the photosynthesis. Can you explain how this conclusion could be drawn? So now the experiment. It is interesting to learn about those simple experiments that lead to gradual development in our understanding of photosynthesis. Chapter you are reading, I have revised this yesterday. Okay, fine. Yeah, I also revised, but I just want to learn the dates and the name of the scientist and the year. So it is possible that this thing is also asked in its exam. Where are you from? I am from where? I am from my home. So Shivam, please subscribe to my channel. That yeah, it may be really revision for you. Yeah, good to hear that your father was also a doctor. Then why are you weeping? Because of the pain of not becoming a doctor? Oh yes. Never trusting HNC. Okay, fine. Joseph Priestley in 1733 and 1804. In 1770, 1770, he performed the experiment. Joseph performed a series of experiments that revealed the essential role of air in growth of green plant. Priestley, you may recall, discovery oxygen in 1774. Priestley observed that a candle burning in closed space, a bell jar, soon get extinguished. Similarly, a mouse would soon suffocate in closed space. He concluded that a burning candle or an animal that breathed the air both somehow damaged the air. But when he placed a mint plant in a same bell jar, he found that the mouse stay alive and the candle continue to burn. Priestley hypothesis. Priestley hypothesis. Hypothesis means he just only thought about it. That the plant restored to the air whatever breathing animal and burning candle remove. Can you imagine how Priestley would have conducted the experiment using a candle and a plant? Remember, he would need to rekindle the candle to test whether it burned after a few days. How many different ways that you can think of light the candle without disturbing the setup? So how many days? Let you think about that. Using a similar setup as one used by Presley, but by placing it once in the dark and once in the sunlight. Jan Indian house. Purify the air, fold by burning candles or breathing animals. Indian house in an elegant experiment with aquatic plant shown that in bright sunlight small bubbles will form around the green parts, while in dark they did not. Later he identified these bubbles to be of oxygen. Hence he shown that it is only the green part of the plant it is only the green part, while in dark they did not. Later he identified these bubbles to be of oxygen. Hence he showed that it is only the green part of the plant that could release oxygen. So this experiment verified that the green plant of the plant would release the oxygen. Now let's turn there and jump on this page. It was found. It was not until about. Okay, fine. The person, please like to my live session. Please like to my live session and subscribe to my channel. It was not until about 1854 that Julius von Zach provided evidence for the production of glucose when plant grow. Glucose is usually stored as starch. Later, studies shown that the green substance in the plant chlorophyll, as we know it now, is located in special bodies, and these special bodies is known as the chlorophyll. And the scientist would not know about the chlorophyll. That why he explained that the plant form a glucose in a special bodies, and this special bodies is known as the chlorophyll, is located in the special bodies which is known as the chloroplast within the plant cell. He found that the green part in plants where glucose is made, and then the glucose is usually stored as starch. Now consider the interesting experiment done by Engelmann. The Engelmann is another scientist who done the experiment. By using a prism, he split light into the spectral components and then illuminated a green alga, cladophora, placed in the suspension of the aerobic bacteria. The bacteria were used to detect the site of oxygen evolution. He observed that the bacteria accumulated mainly in the region of the blue and red light of the split spectrum. A first action spectrum of photosynthesis was thus described. It resembles roughly the absorption photospectra of chlorophyll A and B. By the mid of 19th century, the key features of plant photosynthesis were known, namely that plant would use light energy to make carbohydrate from CO2 and water. The empirical equation representing the total process of photosynthesis for Oxygen evolving organism was then understood as, and you will never pass the exam like this. How do you say this, that I will never pass the exam like this? Yeah, it might be possible just because of my preparation was not so well. I am not giving lots of mock tests. But you did not have right to say anything about me. Just you never pass. And if I pass, then what would you say? Yeah, it may be possible. I just want to recall all the years and the dates and the name of the scientist. That's why I study like this, Mr. KK. Oh, I didn't did it the whole day. It's my first live session in night, and just because of what? Just because of time management. I did not complete the target today, that which I make. What about you? My live session is about only 50 minutes. So after 5 minutes I end it. And then I started. Oh no, it's not my first live session. It's first live session of my at night. Soul of War. क्या है भाई? No need to say sorry, Mr. KK. Anuj, Soul of War, please like my live session that you saw there. Oh, there is no any valid reason for that. Because we both are a neat aspirant and we both understand the pain of each other. That's why. What I thought, Mr. KK. Okay, fine. Then please subscribe to my channel and like my this live session. No, it's not my first exam. I'm a 2+ year dropper. I lost my interest in reading NCRT. That's why I want to interact with some neat aspirant. Yeah, I told you. 2+. What about you? Are you fresher? I'm not fresher. Okay, fine. Oh, are you fresher? I'm studying photosynthesis and half chapter C3, C4 photo respiration I have done, and now I'm read the experiments, and after that oh fine. what about physics and chemistry? I have did it. Tomorrow I show you my short notes that which I make during my preparation time. How many marks you got in NEET 2026? Okay, fine. I have to complete this experiment and then end the live session. CO2 plus water give rise to what? CH2O. CH2O and then oxygen. When the question is arises, then this oxygen come from where? So then the scientists come and perform an experiment. 368, yeah, it is good also in first attempt, very good. When I gave the first attempt, I got 130 marks. Oh, sorry, 131, I think, something like that. And from 131, I switched to 480 in 25. No, 25 nahi, in 24. And then switched to 500 plus, and then again my downfall is started. Hard work, not hard work. It is all about the revision and question practice. Yeah, what I okay, fine then. I'm going to end this session. But it is all about question practice and testing and revision. What I learned in my drop year, that I'm not getting a very good marks. Okay, fine. Thank you all. Please subscribe to my channel if you like it then. Oh, I already read this time. This time I thought this time, or if you subscribe then you get a notification, I think. Then the live session notification come or not, I don't know about it, because I am very well new on this. Okay, fine, yeah. Dramatic 62 I also did. Subscribe. When you come tomorrow, it's not fixed that I will come tomorrow or not. I lost my interest in this book just because of what I don't know also. That's why I used to start the live streaming and want to interact with the NEET aspirants.